Okay, so this PowerPoint is going to be about why living cells need energy and how they make it. So if you think about it, all cells need energy to do a variety of things. Essentially, they need it to be able to grow, reproduce, and repair. An example of a biological process that requires energy is transport of things across the membrane. So in that little animation there, you can see this thing's changing shape and it's causing things to go through. And from time to time, an ATP comes along to donate the energy. ATP is the currency of the cell. It's how the cell pays for all of the things that go on there. So we're going to learn a lot more about ATP over the course of this slideshow. There it goes, happening again. All right, you learn more about that active transport in the next section. So this is probably a bit of a review for you if you've done biology before. Two key terms here are autotrophic or heterotrophic. So organisms can be either autotrophic or heterotrophic. We are heterotrophs and your plants in your garden are all autotrophic. So autotrophic things are things like plants, some protists, and by that algae are the type of protists that are plants. They're green, right? And also some bacteria, and you can see I've got algae here written in like speech marks because blue-green algae is not really algae, it's bacteria. So all of these things make their own chemical energy through the process of photosynthesis, which you should be familiar with. That's why both of these things over here are green. This blob of algae and this basil plant, both green. Now you might be thinking to yourself, how is it that bacteria can do this? Because they don't have chloroplasts in them. And the way that bacteria get around it is they just have chlorophyll, which is the green pigment, inside their cytoplasm. So these guys get all of their energy from the sun. Heterotrophs, on the other hand, hetero means other. They have to get their energy from someone else. They need to take it in by eating something else, usually. Fungi don't eat, but they sit on top of things and dissolve them and start to take in the molecules that way. Most protists, like amoeba, and amoeba are the sorts of things that they're a blobby single-celled organism that can engulf other single-celled organisms. And most bacteria have to take in their materials from their environment as well. They can't actually, most of them cannot photosynthesize. So the sun is the main source of energy for life. We need the sun to provide energy to all of those autotrophs that are out there doing their thing. And so in these 4 pictures, you can see that these plants all growing in the sunlight. There are other energy sources for some things that live on the bottom of the ocean. They get their energy from breaking down chemicals in the rocks, breaking down sulfur in particular, but that's a very small percentage. Most things on the earth get their energy from the sun. That's the ultimate source of energy, whether they make it themselves or they get eaten. Now, photosynthesis, when you've looked at it before, you've come up or you've seen a fairly simple equation, which is carbon dioxide plus water in the presence of chlorophyll takes you to oxygen and glucose. And what I've got here is just something really to show you that it's a lot more complicated than that, and as is common in biology when we teach you something, there are actually lots of little steps that happen. You don't need to know all those steps, but even if we look a bit more closely at this, we can see those 5 things that were mentioned before: light, water, oxygen, glucose, and carbon dioxide. We summarize the equation as water plus carbon dioxide in the presence of light and chlorophyll changes into glucose plus oxygen. You need to remember that. You also need to remember the chemical equation, which is 6H2O plus 6 carbon dioxide goes to C6H12O6 plus 6O2. So those 6's there, if you're a chemistry student, you should be able to balance this out yourself. You know you're going to have 6 carbons over here because there are 6 carbons in glucose. You need to remember that glucose is C6H12O6. It's a carbohydrate. Hydrate. Sorry, my writing's not good on this. And the carb bit is here. the hydro bit is here, and 8 on the end of a chemical thing usually means oxygen in there, okay? So carbohydrate has got those 3 things. You just need to remember 6, 12, 6. Bit of a reminder of where photosynthesis occurs. It occurs in the chloroplast. So we've got the chloroplast up here. Oops, there it is. One there, another one here. So a typical plant cell that's near the surface of the leaf will have lots and lots of chloroplasts in it. And as I said before, in prokaryotes, they don't have a chloroplast, so their chlorophyll, which is the membrane, the pigment that absorbs light and does a few special things with electrons and such, that is just in the cytoplasm for a prokaryote. So prokaryote, as you recall, very simple, much smaller than a eukaryote. They've got a bit of DNA down one end making a loop. They'll have ribosomes around, just sitting there in the cytoplasm. They have little plasmids. Oops. I can't draw little circles. It thinks I'm trying to do right clicking. Anyway, I'm quick. Little plasmids. And also, it's got the chlorophyll just sitting around in the cytoplasm there. It's not actually in the chloroplasts for them. So I'm going to go into a bit more depth with what the structure of a chloroplast is and where this all happens. So if we have a bit of a closer look here, the reactions take place in and around layered stacks of membranes inside the chloroplast where the chlorophyll pigment captures light energy. And let's have a better look at a better diagram of a chloroplast. So here in this chloroplast, you can see that there's the outside membrane, then there's another layer inside that membrane, and then inside there's all of these little membraney bits just here and here and here, and they make these stacks of little disky things. So these individual little units are called thylakoids. Put them in a stack and it's called a granum. A granum? Yeah, granum. That's the singular. Altogether we've got one granum, another granum, another granum. Together that makes grana. Is the plural? Is the plural? And that's the singular. Okay. Look, if you said one or the other in a test, I wouldn't be too fussed if you had to answer that question. So a reminder from the last PowerPoint where we looked at the structure a little bit. Chloroplasts have an external membrane and an internal membrane, which I just showed you over here. Inside that is a further system of internal membranes to increase the surface area available. So this is again about making larger surface area. It's such a common theme in biology. Inside your intestine, you've got a large surface area. Inside your lungs, you've got a large surface area. And even down to the level of cells, we're trying to increase surface area by having lots of membranes. Where photosynthesis actually occurs is really, it's happening in the membrane. Down here, you can see a thylakoid that's been blown up, and there's these little red blobs here. What they are is they're different protein complexes, and they work in they work together. They capture, this is where the chlorophyll is, it captures the light, they pass some electrons around, and in the course of that, what happens is we get a little bit of ATP being made, and this whole process also results in the carbon dioxide being turned into sugar, and we've got the water being split apart and a bit of oxygen coming out. So we've got here the names thylakoids, grana, and the fluid around the stacks is called the stroma. So we've got the here we go, stroma. Just here is the liquid that it's inside it. If inside the cell we'd say cytoplasm, in here it's the stroma. Now a couple of other interesting things we can see in this diagram. We can see they've got their own ribosomes, and they've got their own ring of DNA. And what that means is that actually this is evidence for the theory of endosymbiosis, which you don't know about in this unit, but you might need to know a bit later on in evolution. The theory of endosymbiosis basically says that Chloroplasts and mitochondria were both free-living organisms at some stage before they were engulfed by something bigger and found a new home there. Eventually, this whole system evolved into eukaryotic cells. Okay, so next key idea is that energy transformations are occurring within all living cells. We've got chemical energy turning into kinetic energy. We've got kinetic energy turning into chemical energy, and light energy coming in and turning into chemical energy. All these different transformations occurring. Couple of sort of chemistry type concepts here. One is that breaking bonds requires an input of energy, and another one that creating bonds tends to release energy. So whenever you have a chemical reaction, there is going to be some breaking and some creating. If the energy released by creating bonds is greater than the energy required to break bonds, then there is a net output of energy that is available to do work. And that's what's going on here. Whenever we see a bit of ATP coming along, the ATP drops a phosphorus, or a phosphate group, and that binds with something else. And out of all that reaction, a little bit of energy is available to do stuff. Here we can see a reaction. Should be familiar. This is sugar and oxygen becoming carbon dioxide and water. So that's actually the equation for cell respiration. Okay, doesn't really matter what the reaction is, but it just happens to be that on this diagram. And you can see the difference between this level of energy here and this level of energy down here. That difference there is the amount of energy that's going to be released. A lot of it is lost as heat, but some of it could be available to do some work. While we're thinking about this, what could make this activation energy lower? What would make the graph look more like this? If your answer is an enzyme, you'd be right. Okay, so enzymes can do that. Just as an aside and as a bit of a review. Moving on. Okay, you need to know the ATP ADP cycle. So we've got some ATP here. When the energy needs to be released from that to do something, this phosphate here gets cut off, and whips off somewhere else, and reacts with something else, and there's some energy released, and you've got then ADP. The T stands for tri, which means 3, and the D stands for di, which means 2. So you've got the ADP there, and if you have the right things going on in your cell, a bit of respiration or whatever, then you can add that phosphorus back on to make ATP again. So this should be an example to all of us. It's an example of recycling, where ATP gets used to do something, and then we're left with ADP, and essentially put another phosphate back on, and we've got ATP again. So your cell is it'd be bathed in phosphate groups, and there'd be plenty of ADP floating around, being turned into ATP. Okay, so this is where you might have a couple of questions, because here we say that the bond holding the third phosphate group is unstable and can be broken with the aid of ATPase to release energy. ATPase, the A's, what does that mean? A's means enzyme. Okay, that's really, really messy writing, but I just said enzyme anyway, so you know what I'm saying. That's an enzyme that can make this happen. Now, the text and the diagram above may prompt 2 queries from you. Have a close look at this structure. Does it look familiar to you at all? Does it have anything in there that might look a bit familiar? Let's see what you think. Okay, I've made this bit here flash. Is that the same ribose and adenine as in RNA? Yes, it is. Okay, so ribose from the ribonucleic acid, adenine, one of the bases, it's A. Okay, so this here with the phosphate attached is a nucleotide for adenine, and this sugar sugar, phosphate, backbone, okay? That joins together to make the long strand of the RNA. So yeah, lots of things in the cells have more than one use, and this molecule is handy not only for carrying energy about, but also for making RNA. The other thing is, a couple of slide backs I said that breaking bonds requires energy, yet here it is, releasing energy. Am I lying? Well, it would require some energy to break this bond, but then you've got this phosphate available to go and bond with something else, and this could potentially bond with something else. But the phosphate is actually to do with bonding reactions that occur at the same time that are actually producing the energy. So we would still tend to say though that this bold bit down here, energy is stored in the third phosphate bond and released when the third phosphate is cleaved off. Cleaved means cut. And ATP is converted to ADP. So again, biology is more complicated than we let on a lot of the time. And to get through the Year 12 course, you need to know a little bit of depth, but not a great deal of depth. So just knowing that ATP carries energy, if you break off that last phosphate, you will get energy being released. You need to remember ATP plus water goes to ADP plus phosphate, inorganic phosphate here, that's the I, plus energy. That's the ATP-ADP cycle. Next idea. Explain how most autotrophs and heterotrophs transform chemical energy for use through aerobic respiration, as illustrated by the following equation. C6H12O6 plus 6O6 plus 6CO2 plus 6H2O, which is glucose plus oxygen goes to carbon dioxide plus water. You should be familiar with this if you have done year 11. This is an example of, this is the equation for aerobic respiration. Aerobic means it involves oxygen. So we've got a balanced equation here. You need to remember this. And if you look at it, you'll notice that it is the reverse of photosynthesis. The products over here become the reactants over there. Now, they're not actually exactly opposite to each other. This is a summary reaction. It's summarizing what's happening to this sugar and this oxygen and what we're getting out of it, but it doesn't actually tell the whole story. It doesn't tell you that there are lots of other things going on in between, lots and lots of other steps. Likewise, with the photosynthesis, many steps involved. They're summaries, and it just so happens that they have the reactants here being the products down there, and vice versa. You could also say here that we also got ATP being produced, but it's not actually a product of these reactants over here. So maybe it's better to say, to add on there, and ATP produced as a consequence of this cascade of reactions. Okay, let's have a look at what's next. We're going to break down aerobic respiration into a few different stages. So there are 3 stages that we're going to look at. The first one is called glycolysis. Glyco means sugar, and lysis means splitting. So this word literally means sugar splitting. You need to know that it occurs in the cytoplasm, and it breaks 1 glucose molecule into 2 pyruvate molecules, and there's a net production of 2 ATP. So that's pretty cool, isn't it? You just have 1 glucose and you get 2 ATP out of it. Sounds great, doesn't it? Okay, this is just the first step, and we've already got some ATP. So I've put here a diagram which actually shows you all of the steps in glycolysis. You can see we've got glucose here, and we've got the end product, pyruvate, down here. We know that the cell does things in short steps for a couple of reasons. One reason is that if we were to have a reaction, and we were trying to turn one thing into another, The activation energy would be really huge if we're doing a big change. If we're doing lots of small changes, then we're doing one little reaction. Whoops, this is going to be a problem for me. I'm going to have to do it quickly. One little reaction, and then another little reaction, and then another little reaction. I can probably find a better graph. But essentially, we end up with lots of little bumps. You've seen graphs like that before, where there's just a little bit of energy every time this happens, where we've got that's being released as heat. If we have, I'll just take that back a bit. Only a little bit of activation energy is needed for each of these steps. And as well as that, less heat is released because we've got small amounts of heat at each step rather than a huge amount of heat at the end. You'll also notice that I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 things ending with A's. It's 10 enzymes involved in this process. And so if we think back to DNA and proteins, each of those enzymes requires at least one gene to make it because they're all proteins. And some of them will take more than one gene because they might be a protein that's at the quaternary level of structure, which requires more than one polypeptide. So we've said before there are thousands of genes, and this is one of the reasons we've got thousands of genes, because processes that we often represent in a simple way actually have lots of enzymes involved in them. You can also imagine that if you have a mutation in one of these, the genes that codes for one of these enzymes, that could cause big problems for your body because obviously if you can't do glycolysis, you can't get energy and you would die. So isn't it great that our DNA replication process and our DNA transcription process and translation process is so accurate? Moving on. The second stage of this is called the Krebs cycle, named after Adolf Krebs. Okay, really, really great dude. Discovered it in the 1930s and got the Nobel Prize for it. Well worth it. You might see it written down elsewhere as the citric acid cycle or the TCA cycle. In fact, in this diagram here, it's called the citric acid cycle. That's fine. You can use those names as well. Krebs cycle is in the in your text, and we'll go with that. This occurs in the matrix of the mitochondria. So if we just get into the bottom here, I've got a little bit of a line diagram of what the mitochondria looks like. It's got an outside membrane here. Oops. Outside membrane and an inside membrane with all these folds here. And inside here is the matrix. Okay, into the matrix, just here. That's where the Krebs cycle occurs. You do not need to know this big diagram here, but again, showing you all these words ending with A's, more enzymes involved. More. Okay? And again, just amazing the complexity of what's going on in the cell. The bold stuff up here is what you really need to know. The pyruvate, which we got from glycolysis, here it is coming into this whole process, is converted into acetyl-CoA, and then it goes through the cycle, and it gets broken down into water coming out here. And where's the carbon dioxide? Carbon dioxide will be around somewhere. Oh, here it is, carbon dioxide coming out, carbon dioxide coming out. Okay, so it gets turned into water and carbon dioxide. And as well as that, we end up with protons and electron carriers. That's a proton there. That's an electron carrier and so forth. All of these blue things here, they're important for the next step. You don't need to know what they are, but the fact that this cycle, this is where you get the carbon dioxide and water happening here, and you get numerous protons and electron carriers. And let's see what they're used for. They're used for something called phosphorylation. So there's a little bit of extension here you can get into if you want. It's really quite interesting. It occurs on and in the inner membrane of the mitochondria. It adds a phosphate to ADP to make ATP, and which adds that on, and it makes 34 ATP. So as a result of the work that Krebs cycle did, it didn't make any net ATP yet, but the phosphorylation makes 34, bringing the total to 36 ATP per molecule of glucose. Now, for some reason, the video does not look like it's going to work. I would suggest you watch it later. You've got a copy of this. It shows there the ATPase making the ATP and how it all works. It's really quite fascinating. You don't need to know it all. You just need to know the name of the process, phosphorylation, putting on a phosphorus, that's what it means, where it occurs on and in the inner membrane of the mitochondria. It adds a phosphate to ADP to make ATP, and it makes 34 ATP. So we can summarize that just here. Summary of aerobic respiration. Glycolysis occurs in the cytoplasm, 2 ATP net produced, and 2 pyruvate. I should probably change this, pyruvate. The Krebs cycle occurs in the matrix, supplies electrons to the electron transport chain, and phosphorylation occurs on the inner membrane. 34 ATP are made. This diagram is a really good summary of what's going on here. We've got, hang on, there's a little bit of ATP over there in the Krebs cycle. Sorry. We've got the 2 ATP from glycolysis being made here. This is happening in the cytosol. The cytosol really is a word that is used sometimes interchangeably with cytoplasm. It just means the liquid part of the cytoplasm. We've got the pyruvate coming inside the mitochondria, and we have got Krebs cycle here. They've called it TCA cycle, same thing. You can see it's a cycle, and it's producing carbon dioxide. There's some water coming out. Hang on, the water's not coming out of there. There's carbon dioxide coming out of there. And we've got these things that are called proton carriers. You don't really need to know, or electron carriers, sorry. You don't really need to know much about that. They do stuff on the membrane, which leads to phosphorylation on the membrane. These purple things are ATPase. And like I said, worthwhile looking at that video because it really shows you how they whip out the ATP. They work a lot like a water wheel that would be used in an old-fashioned mill or to cut timber or something. You know, you've got the water in the river running past the building, it's going over a wheel, which is turning machinery inside. This works in a very similar way. These here are all working to put hydrogen ions or protons from the matrix into this area here called the intermembrane space, and they just they tend to go out due to diffusion through these the ATP synthase, and that makes these different. That makes the ATP from that. So it goes beyond, but you do need to know that phosphorylation occurs on the inner membrane. Okay, keep that in mind. Now, not all organisms can do that, and not all organisms can do it all the time. So sometimes we have to do some anaerobic respiration. Anaerobic means without oxygen. That's what the anabit means there. So no oxygen involved. And you can see these two reactions here. No oxygen, just the glucose. In plants and yeasts, that glucose can be converted into ethanol and carbon dioxide. And humans have made use of this through the years. I've got some bread down here. Obviously, this is on repeat, but that bread's rising because the yeast in there, which is a fungus, is breaking down the sugar in the bread and turning it into carbon dioxide and a little bit of alcohol. So don't go home and eat all your bread dough. You would have to eat a lot of bread dough to get drunk. But when it gets cooked, the alcohol evaporates, so you don't actually get any alcohol from it. But that carbon dioxide has made it nice and puffy by making little air bubbles, little carbon dioxide bubbles in it. Over here, the beer as well. Okay, so when you brew beer, you've got yeast in there as well, and that yeast breaks down the sugar that you're brewing, often malt sugar, and it turns it into ethanol and carbon dioxide. And that's where you get bubbles from beers that have been brewed in the bottle. In animals, we thankfully do not make alcohol when we exercise, or we would not be able to do much exercise, would we? Instead, humans make, and other animals make lactic acid. Lactic acid, you would be probably familiar with the name, the words lactic acid. It accumulates in the muscles and it causes fatigue. So eventually, it's acts in a kind of a toxic way, I guess. It makes your muscles sore, and your body can only do so much of this before